It looks doesn't work.
Hello, okay.
Doesn't work.
Hello.
Morning.
Morning. Good morning.
I'm the afternoon. Your mom.
Morning and afternoon. Yes, indeed. So
Hello.
again.
Morning.
Morning. Good morning.
I'm the afternoon. Your mom.
Morning and afternoon. Yes, indeed. So
Hello.
again. Morning. Good morning.
I'm the afternoon. Your mom.
Morning and afternoon. Yes, indeed. So
Hello.
again. Morning. Good morning. Morning and afternoon. Yes. Okay, can you please try now?
Okay, it works. No problem.
Perfect.
Okay.
Okay, can you please try now?
Okay,
This
it
is
works.
for conversation. No,
No problem.
Perfect.
just for the closing ceremony. This is for conversation. No,
No problem.
just for the closing ceremony. This is for conversation. No, just for the closing.
Okay, Marcos, if you are confident, then. Okay, fantastic. Yeah. Okay, Marcos, if you are confident, then.
Oh, this thing also for the power session 1.
Okay, Marcos, if you are confident. Can you please write it on the chat so that he has it written here?
This thing also for the power session 1.
Okay, Marcos, if you are confident. Can you please write it on the chat so that he has it? Yes, this is parallel session 1.
Also
Yeah.
for the power session 1.
Yes, it is for parallel session 1. Yeah.
Can you please write it on the chat so that he has it?
Also the closing
Yes,
session.
this
is parallel session 1.
Also for the power session 1.
Yes, it is for parallel session 1. Yeah.
Can you please write it on the chat? Yes.
Also the closing
Yes,
session.
this
is parallel session 1.
Also for the power session 1.
Yes, it is for parallel session 1. Yeah.
Can you please write it on the chat? Yes.
Also
It's also for
parallel session 1.
closing. Yeah. For the closing session. Yeah. It's also for closing. Yeah. For the closing session. Yeah. It's also for closing session. Yeah.
Good morning.
Morning, Stefano.
Yo no puedo decir que los datos que saqué para el sujeto no sean igual para el sujeto 2.
Sí.
Y entonces, pero en este caso probamos en el que se hizo ahí La tarea normalmente es de esa base de datos, tiene 4 tareas, pero nosotros solo usamos las más básicas acá, que fue mano derecha y mano izquierda, la tarea biclass.
Listo.
O sea, distinguir entre dos nomás, no multiclase para pues evitar, nosotros hacemos eso, pero más adelante. En este caso, la abstract es solo para tratamiento biclass, a derecha e izquierda.
Hello, Dr. Saif.
Hello, Dr. Mahmoud, how are you?
I'm good, thank you. Everything all right?
Yeah, sure.
So I have made you co-host.
Okay.
At the same time, all the presenters who are here in the session, I'm going to make them co-host. Yeah.
Okay.
Okay, so it's 1 past. So please, well, you are the chair of the session.
Okay. All right. Thank you, and welcome to the parallel technical session number 1, track 1, entitled Cognitive and Computational Foundations of Brain Science. My name is Saif Al-Dawlati. I'm an associate professor at Ain Shams University, Egypt, and my co-chair for the session is Professor Muhammad Shahadat Hussain from University of Chittagong, Bangladesh. We will start with the first paper entitled Inferring Neural Circuit Interactions and Neuromodulation from Local Field Potential and Electroencephalogram Measures. The authors are Pablo Martinez Canada, Shahrayar Lowell, and Stefano Panzeri. I'm gonna play the video right now and share my screen.
Hello, my name is Stefano Panzeri from University of Hamburg Medical School and Italian Institute of Technology, and I'm here to present our work entitled Inferring Neural Circuit Interaction and Neuromodulation from Local Field Potential and Electroencephalogram Measures, and this joint work with Shahrayar Lowell and Pablo Martinez Canada. And the paper is about developing computational methods for the analysis of mass signals. Mass signals recorded from the brain are those signals like, for example, electroencephalogram or field potentials or functional magnetic resonance imaging measures that are obtained from the brain and don't have a cellular resolution. They are signals that conflate different neural phenomena. And so the fact that they aggregate data on the one hand makes it, those signals have a high signal to noise ratio, and also being able, for example, to pick very well oscillations and things like that. But at the same time, they conflate many different neural phenomena, and so are difficult to interpret in terms of the actual neurophysiology, the neural processes that generate them. And with our work, with our group, over many years, we have done developed a lot of computational tools based on the principles of information theory and the way physical plausible neural network modeling that I hope helps to some extent understanding and the interpretation of mass neural recordings such as electroencephalogram or field potentials. And in this talk, I'm going to review them very, very quickly. And one thing that we did was to develop computational models together with information theoretic measures, and also then design perturbations inspired by computational work to take a spectrum of field potential and oscillation spectrum in a given brain area, and being able, I think, for the first time to break it down completely in contribution from different neural mechanisms. And so a power spectrum quantifies the amplitude, the square of the amplitude of oscillations from the brain signal recorded at different times. So this is from visual cortex. You can see the same spectrum from the same location, either during stimulation with naturalistic movies or during spontaneous activity. And if you look at this, you see that you have a really broad band set of oscillations. There are oscillations essentially at all different frequencies, from very, very slow, like less than 1 hertz, to more than 150 or so hertz. So they span a very wide range of frequencies. And if you look at them, it's not so easy to find what are the different real bands of frequency region that reflect different neural phenomena. So first, we proposed a computational way to go from just power, so just amplitude of oscillations, to the actual information content and the function that they do. To do that, we developed an information theoretic measure that quantifies how much you can predict which features of the external environment of the natural visual stimulus you can decode at each instant of time from the power of those oscillations. And if we fill that, we find a curve of information carried by the power across frequencies, which is well defined in terms of different bands. And kind of analyzing the information used about further analysis model to disambiguate weight, we were able to demonstrate that in this spectrum you have 3 different regions. One region in low frequencies that encodes temporal movie variations and arises as entrainment of thalamic input to visual cortex. Then a high frequency oscillation in the gamma range that encodes static inverse properties and arises from excitatory inhibitor interaction within the local neural network. And then also a band that does not carry much information about the external environment, but that we were able to impute to neuromodulation, in particular to the variations of dopamine using perturbation experiment that were designed based on the computational work. So in this case, we found that we started with a spectrum that was very difficult to interpret because it was an aggregate signal, and we were able, using various computational techniques, to decompose these into several neural processes, each with a different function in terms of information coding or neuromodulation. And the success that we had in trying to impute different functions and different neural origin to different components of these aggregate signals then led us to think that maybe we could use these model-based computational approaches not only to impute different functions to different parts of the spectrum, but also to use this computational model to estimate the neural processes of what goes on in the brain as we record different components or features of these aggregate neural signals, for example, electroencephalogram of field potentials. And I give you just an overview here what is the logical scheme. So first we study using neural network theory very carefully, analytically and numerically, the relationship between microcircuit neural parameters and EEG or fMRI of potential. Once we've done this very careful study, we subject all this data and our understanding to some model of statistical learning that learn how to associate the various shapes of electroencephalogram spectra and so on to the actual changes in the neural circuit. And then we validate the resulting statistical learning algorithm, their efficacy in estimating the neural parameters using perturbation experiment coupled with the recording of electroencephalograms of potentials and so on. So essentially we ask if the model can correctly detect what is the effect of the perturbation that we know from biological grounds. And finally, once everything is validated, we wish to use these models as an analysis tool to, in quote, invert the electroencephalogram of field potentials of humans in terms of neural parameters, and then being able to estimate, for example, the difference in neural parameters in humans between, for example, illness and healthy brain. In this way, being able to help scientists and doctors to understand better what are the difference in neural processes when the brain is not healthy anymore. And just to give you a very quick review of what those kind of things can do in terms of validation, we did interventional tests or perturbation tests where our colleagues, in particular Federico Rocchi and Alessandro Gozzi at the IIT, and Alessandro Toso and Tobias Donner, some member at UPenn in Hamburg, where they use pharmacological manipulation that change the inhibition excitation ratio. And both in this mouse and the human models, our algorithm was able to predict correctly based only on the spectra of field potential of magnetoencephalographic signals, the actual perturbation work performed. So those methods were able to identify correctly the excitation and inhibition in the actual brains of the neural parameters. And then also we can use those kind of methods also to infer neural computation that would be more useful for basic research. For example, in this application, again, from Tobias Donner and Alessandro Toso's data at the University of Hamburg Medical School, we were able to measure excitation and inhibition ratio over different areas of the cortex and found, as predicted by other models and other evidence, that there is an increase of excitation over inhibition as you go from sensory cortices to more frontal areas. And so in conclusion, and of course I will develop this more in the plenary lecture where I have more time, our work tries to show that computational tools can improve the understanding and interpretation of mass signal recordings, such as electroencephalogram of field potentials, and help identifying interesting components and understanding how these components arise from neurocircuit interactions. And the next step for us are, of course, continue this work to improve our tools to estimate more neuroparameters than the excitation and inhibition ratio, to estimate them more robustly, and so on and so on, so allow people to get a better reconstruction of neurocircuits of mass signals. And in particular, I point to the paper B204 of this conference by Pablo Martinez for recent theoretical work to improve these tools. I thank you very much for your attention. Thank you. Bye-bye.
Thank you very much for your nice presentation. So now I ask the audience, if you have any question, please feel free to ask question to our speaker.
So I actually have one question, if you allow me.
Thank you.
So I can see from the talk that most of the work that you presented in the presentation was focused on EEG and LFP analysis.
Yes.
How about like going one more level down, like going at the level of the single neurons, looking at spike train data? Have you looked at that, and what are your suggestions out of that for finding interactions of neural circuits?
Yes, our model essentially predicts the activity of spike train data, spike train activity, like of classes of neuron, like excitation and inhibition and so on, based on electroencephalogram or field potentials. And here I showed some validation of our method to estimate, essentially based on perturbation of the spiking activity. For example, if you increase the activity of excitatory neuron over inhibitory neurons, or the other way around, but you do it with pharmacological manipulations. But other things that we did, which I didn't show for brevity, is that in some experiments we had data with field potentials and also spikes, action potential of single neurons, and we were able to predict, for example, whether there was an increase or decrease of activity, spiking activity of excitatory neurons, which are the ones that are easiest to record because they're largest. Based on our model. So we did also that kind of validation. We just didn't have time to present it in the talk. So, but we validated the single neuron level. Thank you.
Thank you.
Thank you. So our next paper entitled Intrinsic Motivation to Learn, Action State Representation with Hierarchical Temporal Memory. Now I request the speaker to present their paper.
Okay, I'm going to share my screen for the second talk.
Hello, everyone. My name is Evgeny Cherlikyan. I'm honored to present our team at the conference. We work on biologically plausible algorithms in the Moscow Institute of Physics and Technology at Cognitive Dynamic Systems Laboratory. A paper titled Intrinsic Motivation to Learn Action State Representation with Hierarchical Temporal Memory reflects our recent work on biologically inspired agent models. We have built hierarchical intrinsically motivated agent, HIMA for short, upon the hierarchical temporal memory framework featured by our colleagues from the Numenta company. There is a lot of evidence that successful adaptive behavior requires gaining knowledge from an environment through observation. Therefore, our paper addresses the problem of effective experience accumulation and utilization by an intelligent agent. The most promising results on the problem were yielded by the reinforcement learning field. Despite the recent success of the reinforcement learning due to incorporating deep neural networks with backpropagation error, this framework quickly reaches its limits. The most vivid problem of the deep reinforcement learning is it requires a huge amount of experience. For example, to train AlphaStar to beat a professional player in StarCraft would require more than 500,000 years of real-time experience and accurate simulation of the environment. Therefore, it's not possible to use such an agent in most of the real tasks. As we see, human learns much faster. Therefore, we have a lot of motivation to understand the core principles of human cognition and model it. To build our agent, we have used neurophysiological models of neocortex and basal ganglia. And to model the neocortex, we have used hierarchical temporal memory framework. The model of pyramidal neuron is at the core of the framework. Each neuron has only two states, active and inactive in this framework. Therefore, models work with binary patterns of neuron activation. Hence, the computations are rapid. Numenta has proposed several algorithms that model some neocortex functions. We have used spatial pooler and temporal memory. Spatial pooler encodes dense binary pattern into sparse distributed representation, as SDR for short. SDR has a number of useful features, and there is a lot of evidence from the neurophysiological data that our brain uses the same kind of encoding for internal representations. Temporal memory algorithm enables to learn sequences of patterns, sequences of neural activity patterns. There is a lot of evidence that basal ganglia plays a crucial role in action selection. Therefore, we have built a model of basal ganglia capable of handling sparse distributed representations. Here's the scheme of our model. We divide our input into flows, visual and motor. Therefore, there are two hierarchies, visual on the left and the motor on the right. Let's consider the motor hierarchy first. On its first level, temporal memory learns sequences of elementary action representing raw muscle activity. And on the second level, it learns abstract actions. Abstract action is a sequence of elementary actions. To represent an abstract action in a hierarchy, we use a special naming mechanism that automatically leads to fragmentation of input action sequence into most common subsequences, which then can be reused as an abstract action. To choose sections, we use basal ganglia model. On the first level, it chooses among elementary actions, and on the second, basal ganglia choose among abstract actions. Chosen abstract action increase the probability of choosing a corresponding elementary action. To make a better actions, basal ganglia learns through intrinsic and extrinsic rewards. Extrinsic reward correspond to a vital resource that an agent gets from an environment, and the intrinsic reward corresponds to a prediction error signal that is produced by the first level of visual hierarchy. Let's move to the visual hierarchy now. On the first level, it learns sequences of raw sensory activity, and the second level extracts the most repetitive ones. Visual and motor hierarchies have reciprocal apical connections that help to disambiguate sensory input. Also, apical connections correspond to visual hierarchy projections to the basal ganglia region, which play a crucial role in action selection. To test our agent, we have used mazes on a square grid. Here, agent is depicted as a yellow square, and the goal is depicted as a solid square. The dark blue color represent walls, and shades of bright blue represent the floor. At each time step, agent receives a binary image of a square area around it. Also, it gets a positive reward when it reaches the goal. We tested our agent on different mazes with sizes 8 by 8 and 12 by 12. We compared our agent's performance with the baselines, a random agent and sparse distributed adaptation of Q-learning algorithm with UCB exploration methods. As we see from the figures, our agent converges to an optimal strategy. However, it requires more episodes to converge. It's due to exploration method suboptimality, which is solely based on intrinsic motivation in comparison to optimal UCB exploration method. The main limitations of our research is that our tests are too simple to justify the use of a two-level hierarchy. Also, our visual system doesn't retrieve features from the raw sensory input. Therefore, we can't use this model for more challenging environments than a grid world environment. Advantages of our approach is that due to hierarchical structure of our architecture, our agent is capable of automatic abstraction of automatic formation of abstract actions, and also we have used neurophysiological model of the neocortex to produce intrinsic motivation reward that serves as an exploration motivation for an agent. Also, we have used neurophysiological model of basal ganglia to produce actions and aggregate intrinsic and extrinsic rewards. Our research have shown that the proposed architecture is capable of learning an effective resource searching strategy in grid world environments. Thank you for your attention.
I would be pleased to answer your questions.
Any questions from the audience?
I can ask you a question. So how can you overcome the limitations of your research?
We are going to test our agent on more interesting tasks to justify the use of abstract actions. We are working on this now, and next paper we will publish our results in the next paper. And also we want to add a spatial hierarchy to the visual system to enable extraction of useful features from raw sensory input. Also, we want to add look-ahead planning, which will be based on the internal representational model of the environment, and play with different intrinsic motivation signals. So that's our plans on the future work.
Thank you very much.
Thank you too.
Okay, we'll now move to the third talk today, paper number B28, the effect of expected revenue proportion and social value orientation index on players' behavior in divergent interest tacit coordination games by Dor Mizrahi, Aylan Lofer, Aynan Zuckerman. I'm going to share my screen right now to be presented by Dor Mizrahi.
Hello everyone, my name is Dor and I'm a PhD candidate at the NeuroIS lab at Arena University under the supervision of Dr. Laufer and Dr. Zuckerman. And today I will present the research, the effect of expected revenue proportion and social value orientation index on players' behavior in divergent interest tacit coordination game. Sounds complicated, but we move step by step. So what are we going to talk about today? First, we will go over a brief introduction on tacit coordination game and social value orientation. We talk about research goals, experimental setup, results, and future work. So what is tacit coordination game? Is a game in which two individuals are rewarded for making the same choice from the set of alternatives, where the communication between them is not possible or not allowed. Such an example to such game is the prisoner's dilemma. A divergent interest tacit coordination game is a subgroup of tacit coordination game in which an agreed upon solution might yield a different payoff to each player. So let's give an example to a tacit coordination game. Two people are in different rooms and cannot communicate with each other. 10 single $1 bills placed on each table. The task of each player is divide these $10 bills into two piles. If both players divided the pile by the same way, they will both win the game and get a prize, the same prize. If we look at this problem using the game theory analysis, there is no preference to each one of the different solutions because this is a game with a multiple Nash equilibrium. But if we have to pick one specific solution, most likely 80% to be exact, will choose the equal deviation, 5 bills per each pile. Thomas Schelling proved this point or make this point clear that people can coordinate better than game theory using focal points. Focal points are prominent solutions which detects the player to choose them, which don't take part in the game theory, and this is why game theory does not fit to tacit coordination game. Thomas Schelling won a Nobel Prize on his work in this field in 2005, and he is the first one to define this problem and the solution to this problem. So let's move to the divergent interest tacit coordination game. In this case, the profit of the distribution of the two players have to divide the two piles, but in this case, each pile is represent each player. Pile 1 to player 1 and pile 2 to player 2. If the two players divided equally, each player will receive the amount of dollars in his pile. We understand that now, when the payoff is not equal, there are more considerations that have to be taken into account while the players decide. This game can be even more complicated when there is non-coordination penalty. They will think twice how they allocate their money or the $1 bills. So this is an example to divergent interest tacit coordination game. Let's talk about social value orientation. So social value orientation is a theory that describes the preference or motivation of decision makers. Most SVO questionnaire uses the problem by asking the player allocating joint resources between himself and another unknown player. Most of questionnaire uses 4 main tags: altruistic, prosocial, individualistic, and competitive. But in this study, we used the most advanced method, the SVO slider, which describes the player preference according to a continuous index, the SVO angle, which is highly correlated to the original tags. We can see that an angle of 45 degrees is a prosocial, and 0 degrees is individualistic, 90 degrees altruistic, and etc. And this is done using 6 different questions. In this case, an angle of 38 degrees is a tendency to prosocial, between prosocial and individualistic. It is more accurate method. So in our research, we measure the SVO index of each player. So what are the research goals? The goal of this study was to examine the effect of 2 key variables, the expected revenue proportion that we will define later, and the social value orientation on the probability of realizing a focal point solution in divergent interest static coordination game. Of course, this experiment is a preliminary behavioral examination of decision makers, but we have the intention of confirming that model using electrophysiological measures such as EEG recording. So this is the research goals. And we will select the bargaining table game in our case. It's a well-known game which appeared in different researches. And this is a task and coordination game which consists of 9-on-9 square board. Discs are scattered around the board when each disc has a numeric value. Alongside the disc, there are 2 squares, blue for player 1 and orange for player 2. In our case, all players were the blue square, player 1. The task of each player is to assign each of the discs to one of the squares. So the potential payoff of each player is the total sum of the numeric value of all discs which are allocated to him, to the blue square. In practice, the points of each player receive are only the disc and the both players coordinated on. The blue player gave to himself and the orange player gave to the blue player, and etc., and vice versa. On the uncoordinated disc, both players receive a penalty. We define a 20% penalty of each disc's value. Previous studies, as you can see on the full text, has shown that the closeness rule is the main focal point solution in this kind of game. Closeness is allocating the disc to the closest square, such as simple. So let's talk briefly about the experiment design. Each participant played the same set of 10 different bargaining table coordination games. 4 games as a dominant player. When in focal point solution, he earns more than 50% of the points. 4 games a weakness, which he earns less than 50%, and 2 games of equality when he earns exact 50%. And if the players take the focal point solution in all 10 games, it will accumulate 52 points, potentially. A penalty for non-coordination we talked about, 20%, and all the players were the blue player. We have 70 participants, 34 females, 36 male, with the mean age of 23.5, with a standard deviation of 274. So let's move on to the results. First, we want to find the effect of SVO on the probability of realizing focal point solution. In this section, we attempt to quantify the effect of the SVO index on the probability of the focal point solution realization. We have grouped the experimental results according to the player's measured SVO with a resolution of 5 degrees. And within each group of 5 degrees, we calculated the mean focal point solution probability of the experimental results. We can see using this nice graph of results that we use a linear regression, which was significant in a linear model. And as the SVO angle increased, the greater the chance of realizing a focal point solution. And this makes sense because higher SVOs, the player tend to be more prosocial. Prosocial player would like to share his results for revenues, but we prove it mathematically. Now it's significant and confirmed. You can see the equation of the model and the p-value and all other statistical measures. We go back and do the same for the ERP. What is ERP? This is the expected revenue proportion that reflects the percentage of points that the player receives if both players implement a focal point solution according to the closeness rules. Again, we aggregated experimental results according to resolution of 10% or 0.1. And within each group, we calculated the mean focal point solution probability, which carry out from the experimental results. As we can see on the graph, data analysis reveals that the extreme situations of ERP, when ERP is 0.1 or 0.9, when one player receives large amount of points relatively to the other one, these are excluded from the data because these are anomalies. And the all regular data between 0.2 and 0.8, as you can see in the red dots, the relationship was a significant using quadratic model in a linear regression. The farther we move from the point of equality, which is 0.5, the lower the probability of realizing focal point solution decrease quadratically. So as we move far away from the 0.5, we go down in the probabilities. Finally, in order to test whether there is an interaction between the two variables, SVO and ERP, regarding the effect on the probability of realizing a focal point, we use a two-way ANOVA model. We converted the continuous data into a quantized one, which gave us 3x3 ANOVA array. As we can see, the ERP below and upper to 0.5, and the SVO between 15 and 30, and below and under, and we get a 3x3 ANOVA array. The results of the ANOVA model showed that there was a significant mean effect for the ERP and for the SVO, which is not surprised because we see the linear regression models before, but there is also interaction effect, which was also significant, as we can see on the results. We can see the interaction plot, which there is a different behavior for different ERP by different SVO. The meanings and the full results of the interaction can be found in the full text. We analyzed and we talk about the meaning of each one. Which future work we can do? We want to explore the behavioral differences of the SVO and the ERP, and want to see if they're accompanied by parallel changes in the electrophysiological markers such as theta beta ratio or other cognitive load measures. We want to do a group level, individual level analysis because this is a group level analysis, and we want to combine the SVO and ERP into an autonomous agent who will optimize the divergent interest and coordination games. So I will be happy to further discuss and collaborate. Thank you. You can see my email. You can see the email of my doctors and supervisors and the website of our lab. Thank you very much.
Hello. I will be happy to have questions about the presentation. B208, we just heard.
Any questions from the audience?
I have a question. Could you please elaborate the cognitive aspect of your research a bit more?
Okay. So we have studied the table bargaining game, in which two players have to actually allocate the rewards differently for each other. Now they have to, the aim of the game is to similarly allocate the value of the discs, without any communication whatsoever between the players. This is why it is called tacit coordination game. So actually when there is a penalty, what is the penalty that was mentioned in the presentation? The penalty is when the two players fail to coordinate similarly. Okay, when they do not converge on the same solution, they do not coordinate similarly, and then the penalty of 20% of the value of each disk, they allocate differently for each other. Now, we want the overarching aim of the study was actually to explore two main parameters that can influence the strategy of the players. So one of them is the ERP. It means the potential revenue each player could receive after coordinating with the opponent. And the other parameter was the SVO, the social value orientation, which means whether you are a competitive player or a altruistic player, or you lean towards cooperation. So the SVO actually defines your basic social orientation towards cooperation or competitiveness. So we studied these two parameters, SVO, social value orientation, and the revenue proportion, the potential revenue each player could receive after playing each game. And the graphs that were presented actually display the effect of ERP, the revenue proportion, and of the SVO. We actually seen that there is an interaction between these two parameters, SVO and ERP, so that players which are leaning towards cooperation don't mind much if they win or lose. They are inclined to make a focal point solution. So they won't allocate the money for themselves, the value of the disk to themselves. They would like to distribute it as evenly as possible between themselves and the other player, regardless of whether they are potentially winning or losing the game. So this is the main result.
Thank you very much.
Thank you. Thank you.
So if there is no question, so we can ask the next presenters of the paper. So it is on the extraction of high-level visual features from lateral geniculate nuclear activity, a RET study, will be presented by Megamin, German University in Cairo, Egypt, Islam Monia, Shams University, Cairo, and our another chair of these sessions, Dr. Said Al-Berti. I request them to present their paper.
Thank you. I'm gonna share my screen.
Hello everyone, I am Maya Gamal, a PhD student at the German University in Cairo, and today I'll be presenting the work of our paper titled On the Extraction of High-Level Visual Features from Lateral Geniculate Nucleus Activity, a RAT study. What is the lateral geniculate nucleus, the LGN? As we can see from this figure, the LGN is a subcortical structure in the thalamus. It carries visual information from the retina to the primary visual cortex. The LGN has been known to have a simple linear role as an early stage of visual processing. However, multiple studies have shown that it has a more complex role in visual processing with various nonlinearities. So beyond its known role in low-level vision, the LGN is argued to have a role in higher-level cognitive processing. Accordingly, the aim of our study is to examine predicting high-level visual features from rat LGN firing activity. So what are the high-level visual features we examine in our study? In our work, we have used checkerboard patterns with 4 white pixels in each pattern. So for the first visual feature, we examined the positions of the majority of white pixels in a pattern. As we can see, for example, in this pattern, the majority of the white pixels is in the upper half of the pattern. However, in this pattern, we can see that the majority of the white pixels is in the bottom half of the pattern. For the second visual feature, we examine the scatter of the white pixels in a pattern, which means how far or close are the white pixels to each other. So for example, we can see in this pattern that the 4 white pixels are close to each other. However, in this pattern, there is a white pixel that is far from the other white pixels. We consider these visual features as high-level features because they are global features related to the whole image, not local features related to single pixels in the image. For our data recording, we recorded extracellular neural activity of LGN neurons from 6 female albino rats using 32-channel multi-electrode arrays. We have shown to the rats stimulus that included 4x8 checkerboard visual patterns, as we have shown previously. A total of 32 different patterns were presented 100 times each, and for each pattern, the 4 flickered pixels were on, which means that they were white for 200 milliseconds. So as we can see in this figure, this means that the stimulus was on for 200 milliseconds, and then it was off for 300 milliseconds, in which all the pixels were black. For our data pre-processing, after recording the data, we did a step of spike detection and sorting to extract the spike trains of the neurons from which we have recorded the data. And then we did a step of firing rate calculation to calculate the firing rates of these neurons. We calculated the firing rates by counting the number of spikes in 50 millisecond windows, and then we averaged the counted spikes of each pattern for each neuron across the 100 trials. So for our first visual feature, we examined this feature in the form of a classification task in which the position of the majority of white pixels in a visual pattern is classified into one of 3 classes. The first class is the up class, and in this class, as we can see, the majority of the white pixels is in the upper half of the pattern. The second class is the down class, and in this class, as we can see, the majority of the white pixels is in the bottom half of the pattern. And finally, in the third class, the equal class, the number of white pixels in the upper half is equal to the number of the white pixels in the bottom half of the pattern. For the classification methodology we used in predicting our first visual feature, we compared 3 different types of inputs. The first type is the visual patterns, and in this type we used the raw patterns or the images in predicting the visual feature. And the second type is the single neuron firing activity, and in this type we have used the firing activity of each single neuron separately in predicting the visual feature. And finally, the third type is the population firing activity, and in this type we have used the firing activity of all the neurons in the population to predict the visual feature. We can see from these 3 different types that the first type is different than the other 2 types, in which in the first type we use the raw images to predict the visual feature. However, in the other 2 types we have used the LGN firing rate to predict the visual feature. So for each type, we have built a visual feature detection model. The first step of the model is applying dimensionality reduction using principal component analysis, the PCA. And then we use the reduced dimensions as input to a classifier. We have used 3 different classifiers for comparison, namely the support vector machine, naive Bayes, and linear discriminant analysis. After building the classification model, we have evaluated its accuracy using the macro F1 score metric, and we have used the leave one out cross-validation approach for accuracy evaluation. For the second visual feature in our study, we examined it in the form of a regression task for estimating what we call the scatter index. The scatter index is a metric that quantifies the scatter of the white pixels in a checkerboard pattern using this equation. So what does this equation do? As we can see here in this figure, this equation first specifies the positions of the 4 white pixels in the pattern, and then it calculates the mean of these 4 white pixels, and then it calculates the Euclidean distance between the mean and the 4 white pixels. Finally, it averages these 4 distances. For the regression methodology we used for predicting our second visual feature, we again, as we have shown in the classification methodology, we compared 3 different input types, and for each input type we have built a visual feature detection model. The first step in the model is applying the PCA for dimensionality reduction, and then we use the reduced dimensions as input to a linear regression model. After building the linear regression model, we evaluate its accuracy using the root mean square error matrix, and again we have used the leave one out cross validation approach for accuracy evaluation. So for the classification results of predicting our first visual feature, we can see from this figure the F1 scores achieved across the 3 different classifiers we have used for comparison and across the 3 different input types we have previously mentioned. We can see from the figure that the population activity achieved almost the best accuracy compared to the other 2 types, the other 2 input types across the 3 different classifiers. And we can see that the population activity achieved the best classification accuracy using the SVM classifier with F1 score of 0.67. We can also note from this figure that the accuracies for the single neuron activity and the population activity are averaged across the 6 subjects used in this study. So we can conclude from this figure that the population activity outperformed the single neuron activity, and it also outperformed the input patterns in predicting the first visual feature. For the second figure here, we can see the population feature space of a sample subject across the first 3 principal components after applying the PCA. Each point in this figure represents a pattern, and these points are colored according to their classes. So we can see from this figure that the population activity had the ability to separate the 3 classes using just the first 3 principal components. So we can conclude from these 2 figures that the population activity had a better ability to predict the first high-level visual feature compared to the raw input patterns shown to the rats, and also compared to the single neuron activity. For the regression results, we can see from this figure that the population activity again had the best regression accuracy in predicting the scatter index with root mean square error of 0.3. So we can see again, as we have seen in the classification results of the first visual feature, that the population activity outperformed the single neuron activity and the raw input patterns in predicting the second visual feature. And we can see from this second figure the actual scatter index versus the scatter index predicted using the population activity of one sample subject. We can see the high correlation between the actual and the predicted scatter index from the figure. Each point in this figure represents a pattern, and we can see that the fitted regression line between the actual and the predicted scatter index has an R squared of 0.69. So we can conclude from this 2 figures that the population activity again had a better ability in predicting the second visual feature, the scatter index, compared to the single neuron activity, and also compared to the raw input patterns used in the experiments. So these results again confirms the results that we have shown in the classification task. Then we did performance analysis to our results, and we can see from this first figure the PCA loadings of the population firing rate. These PCA loadings are averaged and normalized across the 6 subjects. We can see the loadings across the 10 time bins of our stimulus, and we can see that the stimulus onset happens at the first time bin and the stimulus offset happens at the 5th time bin. We can conclude from this figure that the first 2 time bins have the highest contribution to the performance achieved in both the classification and regression tasks. And this second figure shows the accuracies of the individual neurons achieved in the regression and the classification task. We can see that in this figure, each point represents 1 neuron from all the 6 subjects. So we can conclude from this figure that there is no correlation between the classification and regression accuracies achieved by single neurons. And this figure also confirms the results we have seen in which the best accuracies could be achieved due to the coordination between the neurons in the form of the population firing rate more than the performance of the individual neurons. Finally, to summarize our results, we first demonstrated the success of the LGN firing activity in predicting two high-level visual features from checkerboard visual stimulation patterns. We then showed that the LGN population firing activity outperformed the raw input visual patterns and the single neuron activity in extracting the two visual features. So these results indicate that the LGN neural activity could contribute to higher order visual processing beyond its traditional nonlinear role. Thank you so much for listening, and I'm happy to receive your questions.
Great. I'm happy to receive any of the questions.
Any questions from the audience? Any questions?
I have a question. That is, in the pre-processing phase, you have used the principal component analysis. Is there any other process that you can be considered which will allow you to give better result?
Yeah, for in the classification and the regression tasks, we have picked the number of principal components that achieve the best accuracy. So we have examined multiple number of principal components to check which number is the best.
What is the unique contribution of this research?
Sorry?
What is the unique contribution of your research?
The unique contribution we consider is that we show evidence that the LGN could contribute to high level visual processing, and this is a direction that is tested now due to the previous studies that have shown that the LGN is just a relay point between the retina and the visual cortex, and it just do some linear processing. However, a lot of studies have shown that it could contribute to more higher level processing, and this what our paper is trying to show, that it has some contribution to high level processing, visual processing, through the two-level visual feature we have shown.
Thank you very much.
Thank you.
Thank you. We'll now move to the next paper, number B211. The title of the paper is Disfluency as an Indicator of Cognitive Communication Disorder Through Learning Methods by Aurelio Lopez Lopez and Marisol Roldan Palacios. It will be presented by Marisol Roldan Palacios. I'm going to share the presentation right now.
Good morning, good afternoon. We are presenting the work Disfluency as an Indicator of Cognitive Communication Disorder Through Learning Methods. The authors, my supervisor, Aurelio Lopez Lopez, and me, Marisol Roldan Palacios. We start explaining the subject of the study. Then the steps followed in the methodology are described. The selected feature is presented. We follow with a brief discussion of the results. A revision of the complexity of the subject of study is added. A few of detail is given about the longitudinal model, and conclusion and further work are delineated. There are a variety of causes that can produce different adjustments in language. We're working on transcriptions of short narratives of Cinderella routine tasks taken from participants being affected by a traumatic brain injury. The samples correspond to the first year of recovery. The type of variables analyzed are fluency indices. The methodology starts with a principal component analysis to determine the features involved within the linear relations of the components and in the major variance. In our context, we found that 12 principal components contain more than the 90% of the variance, as the elbow graph can show. Then the correlation among those attributes is revised, and the less correlated are candidates to be evaluated with learning methods to place the most indicative to a discriminative task. The cycle is repeated until we identify the feature subset going beyond the score achieved by the baseline set. The described procedure brings us to determine a subset of 4 features. We have the utterance densities at morphological level, the average of the repeated units, and the total of the phrase repetition, in addition to the proportion of the word revisions. The mean of repeated units is calculated by this rule. It divides the sum of the part word repetition minus repeated units plus the whole word repetition minus repeated units. And it is divided between the addition of these same 2 variables without resting the repeated units. One finding in this point is that attributes associated with typical disfluency or those related to stutter-like disfluencies, when evaluated alongside other features, are adding a noisy effect, though some of them seem to contribute information. Here are included the results from 2 methods based on 3, the random forest and the Spark. Besides, for comparative purposes, naive Bayes working on likelihood calculation is considered too. The manner we read the curves is the dotted lines correspond to the baseline, and the continuous are the curves of the 4 feature subset response. On the x-axis is running the evaluated period, and on the y-axis is reported the F1 macro score. We see that the curves of the selected characteristic remain above of those of the baseline for each algorithm. The baseline was calculated evaluating the entire fluency variables and taking the highest score from any of the used learning methods. The score achieved by the 4 feature subset condensed the total of the information contained in the baseline set and is good enough for labeling the feature instances. The time, they are also sensitive enough to reflect subtle changes from one time point to the next. Generally, we see that naive Bayes replicates the answer from the methods based on trees. So the scores move in lower levels. All of the methods were evaluated with leave one out cross-validation. This criteria is convenient due to we are handling sets of 42 instances in average for each group and per time point. To gain comprehension about the complexity on the subject in hands, we put together in a graph the individual indices of each participant per selected variable. On the left side, we have the behavior of the study group, and on the right side are the trajectories for the group of control. The curves correspond to the proportion of world visions, where we can identify at least 3 patterns. The first, in appearance steady, lives below 0.25. The second is a bunch of curves alternating valleys and peaks. And the third is a cluster of trajectories growing during the first 3 recovery stages to then drop for the last one. Once we covered the first objective of identifying a subset removing redundancy, performing properly indicating the changes in the response of the language affected by a traumatic brain injury, the task naturally follows the revision of a predictive ability that we tried with mixed effect models. However, the evaluation of the characteristics for this modeling was not direct. We found some singularities generated in the proof of singular significance of the model. Among the conclusions and further work, we have that fluency attributes are good indicators for tracing the evolution of the modification presented within a language affected by a traumatic brain injury. That F1 macros cultural learning methods leads the identification of the 4 feature subset within an interval of 90.70 to 92.30 for random forest. That Spark was not far from random forest result, with score leaving few below of those of it. And depending on the purpose of the examination, we found that with less features, it's possible to monitor the affectation of the language that we analyzed. We need to revise in advance and simultaneously the condition to enable the feeding of other instruments and as mixed models with the output from learning methods evaluation. Then we can reevaluate the selected feature subset to have our longitudinal model. We want to recognize the support from CONACYT, Consejo Nacional de Ciencia y Tecnología en México, and to Brain Injury Conference sponsors. Thank you to Instituto Nacional de Astrofísica Óptica y Electrónica and to the Language Technology Labs of the Institute. Thank you for your attention.
Any questions from the audience? Okay, I have one question about the data size. Can you give us more information about the data size, the number of patients, number of samples?
Yes. While we work on samples that in this case is not too small. We are talking about 40, in average, 42 samples per group, per the participants suffering of a brain injury and the control group.
Okay.
Well, in the context of our study, that is not so reduced a sample, because it's hard to get this type of instances.
Yeah, sure. Okay, thank you. Any more questions?
Thank you.
Okay, I think we can move to the next talk. Professor Muhammad, please.
Thank you. Our next paper entitled Directed Information Flow in the High-Level Visual Context. Now I invite the speaker, researcher Piang Li from University of Valencia, Spain, to present research.
Okay, I'm sharing my screen for the next talk.
Today's my topic titles is by directed image flow in the high-level brain cortex. My name is Piang Li. So the first we want to know what the scientific research problems about this research. The first we should know the ways of image flows between neurons. There are three possible ways of neural image flow. The figure A shows image flows from A neuron to B neuron. The figure B shows information flows from B neurons to A neuron. And the figure C shows bidirectional image flows between A and B. That's we are very interested in figure C, how the information flows bidirectional, not just one way from A to B neurons or from B neurons to A neurons. Before we start, we should know some basic information theoretical methods in neuroscience research. The first is entropy, that is undirected. The entropy's definition by selective the summer's probability distribution times log base 2 probability distribution. And then the second is the second where's famous definition is mutual information. It also is undirected, and it's very widely used in the function collectivity research. Now, it's many scenarios how much information shared between variables or between neurons, and each definition buys each of x neuron plus then each of is y neurons, and then minus the shared information between x and y. The third is transfer information. It's directed, but it's just one way or one direction. You can mirror from the information flows from x sequence to y sequence, or from y sequence to x sequence. So the transfer information can be mirrored. There's causal factors between x sequence and y sequence. That means we can predictions next states by we can predictions next states of x sequence by conditioned on the y sequence and the history, the past history of x sequence. Next, we should talk about some methods where used in this research is directed information. We can call DI, which is proposed by James Massey, and it's a very classical definition. And the first, it can be mirrored three information flows, three ways information flow. The first is feed-forward information flow from X sequence to Y sequence. And it also can be mirrored the reverse informations from the Y sequence to X sequence. And then if we know the feed-forward information and the reverse information, we can use definitions. There's a resonance or instantaneous influence. That is the feed-forward information minus the reverse information. And then we can be mirrored by directed information flows with directed information, and that's we are very interested in neuroscience, and it can help us understand how the information flows, specifically by directional information flows in the neurons. So how to say estimate the directed information? We use the setup context tree with approach we can call CTW. It's a lossless cooperation algorithms, and there is this cooperation algorithm better than traditional and commerce will know something very classical cooperations algorithms such as ZIP and SafeZ and theory.gz. And so what's the difference between CTW and others traditional cooperation algorithm? That if we know some sequence, some context sequence X, and then we know the probability distribution of this sequence, and then we can cooperations, whereas we can get with beta cooperations this sequence. But the problem is if we don't know the source of this probability distributions, the sequence, and how we can use cooperations in sequence, and how we can first estimate the source of probability distribution. And CTW can help us to do this. So this figure shows something context tree. If you know the cooperations algorithm, something like half mass coding, and you will better understand the mechanisms of CTW. But because time's reasons, I am not giving you details of the CTW, but you can read this paper by Williams, and it's various lines and various details introduced to what is context trees, weightings, what's the probabilities of CTW, and I strongly suggest that you should read this paper. Then if we have, and if we already know the scientific problems, what we want to do, and then we also have the algorithms, we have tools that we need to data set. That's the third really important. We use Border 5000, this open source data set. It shows some latest images from the citizens, ImageLab at Coco, and then shows these images to 4 subjects, and at the same time it's recordings with activities with MRI, and then after the process and statistics, we can go to something. We can see definition or selection or reconstructions ROI, and then we can have lefters V1, right V1, and we have OPA, PPA, AOC, and ISC, and the Border 5000, you can explore this data set from here, and it's open source. So the 5 is the results. The first thing shows collectivities metrics by Pearson correlation, and the second shows by mutual information, and this is transfer information, just one direction. And we can see that mutual information can capture most rich information, captures rich information than Pearson correlation, and but basic basically, these three approaches all capture something. They have all captured something very important just collectivity, such as from RAC to LOC, from RAC to RLOC. Actually, this is something very important to the function collectivity. These approaches all capture these features. So, like this else RAC and between R's PPA. And this figure shows function collectivity was estimated by the directed information figure. The first figure shows feed-forward collectivity, and the sixth shows resonance information flows between RIs, and the third figure shows reverse information flows. So what we found, first we found that there have information flows within the season selective areas such as OPA, PPA, and RAC. The same thing we also found that information flows between the season selective areas such as RAC and the objective selective areas LRC. The third, we found that there has weak reverse directed information flows in the high level swing cortex. The findings, we should mention some say problems, some limitations of this research, or some say point you should cares, first cares. The first is binary input when, because CTWs, this corporation's algorithms only sees the inputs when you must be the binaries when you, that means you should convert a spotter signal to the binary when it's 0 or 1s. Then how do you properly convert spotter signals? That's very important. The signal is parameters because the CW's estimators here have some say freeze parameters such as the depth of the tree. How to use properly choose these parameters also will affect the final result. The third point I should mention is effect size and powers. That means for such as the width or ROIs, reconstructions or selections. So if you have the different way to selection ROIs, maybe you can get more accuracy or no worse results. So it's depending on the width or ROI selections, and you should be very careful at this point because effect size and powers get us more and more citations recently in the fMRI research. So the finance reference, I strongly suggest that you should read this if you're interested in this. If you're interested in this research, you should read the first reference, causality, feedback, and directed image from the James Macy, and also the second universal estimations or directed information. And the third reference is conversations of material information and directed information. And this reference very simplified and classical paper. The finance is I want to thank something I call it thanks grant spotters this research. Thank you so much.
The audience, if you have any question, please.
So I have one question about the measure used to measure the information flow. So is it capable of identifying nonlinear interactions as well as linear, or it only captures linear relationships? we cannot hear you. So is your microphone on? We still cannot hear you. Kang? So it seems that you might have some audio problem because we cannot hear you. Okay. In that case, I think we have to move to the next talk. It's paper B214, titled Computational Network Model for Shared Mental Models in Hospital Operation Rooms. The authors are Laila Van Mens, Jan Troer, Jan Klein, and Peter Roilovsma. And the paper will be presented by Laila Van Mens. I'm going to share my screen right now.
Hello, everyone. My name is Laila, and I will be presenting the paper, a computational network model for shared mental models in hospital operation rooms. And this work has been done by myself, Professor Jan Troer, Jan Klein, and Peter Roilovsma. I'll first quickly go over what I'm discussing in this presentation. So we kind of are combining two domains, like an approach and a domain. We are using the computational network modeling approach that is developed by Jan Troer, and we are applying this to the domain of intubation, the intubation procedure, which is normally done by a doctor and a nurse, or possibly more medical personnel. So I'll first quickly go over what the domain looks like. So most of you probably have seen this in like a movie or something, but when a patient cannot breathe well, he or she needs to be intubated, and there are multiple ways to intubate. The one with the tube that you're seeing in the picture in my slides, it's called laryngoscopy. And there is also intubations where there's a cut made in the neck of the patient, but that's, for example, this one doesn't work. So our scenario is that a doctor, denoted by D, is called to perform a tracheal intubation, which is the one with the tube that you're seeing, in collaboration with a nurse, noted with the N. So we can divide the operation in a few roles for the doctor and a nurse. So the doctor's roles are the team leader, being the team leader, preparing the team and preparing the team for difficulties, and being the intubator. And the nurse has as her or his roles intubator assistant, preparing the patient, preparing the equipment, preparing the drugs, and then also giving the drugs before the procedure, monitoring the patient, and then giving cricoid force, which is a kind of pressure on the throat of the patient so that the doctor can see in the mouth and can put the tube in, and if necessary, running for help and running for equipment. So together, these roles form a shared mental model because there is more than one person that has a mental model about has roles, and they're basically having a shared mental model both about their own and the other person's roles. yeah, so these are the rules, and now we will go over the steps of the intubation procedure. So let's say a patient comes in that needs to be intubated, and imagine the situation is quite stressful because the patient cannot breathe. So first, the nurse prepares. So these steps, by the way, are from like medical literature, and this is kind of how an intubation should go by the book. So step one, the nurse prepares the patient and performs the preparation of the equipment. This includes the so-called capnograph that we will see later on in steps as well. Then the nurse performs the preparation of the drugs. After this, the doctor starts with the pre-oxygenation of the patient. It means that the patient gets an oxygen mask on. Then the doctor starts with the preparation of the team and the preparation for difficulties. So the doctor will be saying to the team what they're going to be doing, and if something goes wrong, what they will be doing then. The nurse gives the patient the prepared drugs as the next step, and then the nurse starts performing the cricoid force. Then the doctor initiates the executing of Plan A laryngoscopy, which was the intubation that we saw in the picture before. And the doctor kind of announces here that they're starting that. Then the doctor actually starts the first intubation attempt, and the nurse assists the doctor in the intubation attempt. And then the nurse monitors the patient. And when the first attempt is finished, the nurse seeks confirmation of the attempt's success or not by monitoring the capnograph, which the nurse prepared in step one. If the capnograph shows that the attempt has succeeded, then the nurse will verbalize this to the doctor, and the doctor will also verbalize that it has succeeded. So there's like a lot of communication going on here. Okay, so as I said, we are applying the domain to our network modeling approach. So this is the network model. This is like a bigger overview, and you can kind of divide it in three parts: the red, the yellow, and the blue. So first of all, the blue is what is actually happening in the world. So you can see, let me go a bit closer. You can see WS for the blue, which means world state, so what is happening in the world. So this whole line of blue dots is kind of all the steps that we discussed in the previous slide. So the steps that have to happen for an intubation. So you see in the beginning, it says call intub. They're all a bit shortened, so it would fit better. So call for intubation. The next one is prep PN. So that means prepare patient by the nurse. And then prep EC N. So that means prepare equipment by the nurse, and so forth. I won't go over all of them now. So that's like that middle blue part. And that means that these things are actually happening in the world. On top, you see that red circled part. So the circled part is what is happening in the mental model of the doctor. So these steps are virtually the same as the steps in the world states, but they happen in the head of the doctor. So you kind of see the same steps, and you see that it's denoted by DS, so doctor states. And then on the bottom part, you see NS states in the yellow part, and also the circle part is what is happening in the mental model of the nurse. So they're all pretty similar. However, the difference is in those little lighter colored yellow and red circles. These are the ownership states. So as you can see here, OS2 prep PN. These ownership states are the states that kind of make things actually happen. So they only exist for the steps of the nurse and the doctor for the actions that each of them actually has to do. So here it's easy to see kind of the sequence of who's doing what and when. So there's a bit of gaps in there. You see that the nurse is like preparing the patient, the equipment and the drugs, and then the doctor takes over with the pre-oxygenation and team and the difficulties, etc. So you can see it's quite complicated to have this all go smoothly under a stressful situation. And that each of them is actually doing what they're supposed to do and taking over from the other when they're done. In all the way on the right, you see like a bit more states, and that I'll actually zoom in again. So you see that after WS13 over here, observing the Kaplan graph, the nurse actually has to say if the attempt has succeeded, verbalizing success by the nurse, or failed. And this then also goes on for the doctor if they're verbalizing for success or failure. These come from the ownership states that also have dotted negative influence lines. So verbalized failure ownership states from the nurse will negatively influence the verbalization, hard word to say, for success for the nurse. So this is kind of how all these things work. Then I'll just quickly go over our context state over here. That's basically, right now, that's working as stress. So here you can see the whole thing. It is connected to the verbalization of the failure and the success of the nurse. The higher the stress, the higher the influence on the verbalization of the failure. So that means that this kind of shows whether the attempt has succeeded or failed. And currently this is not really dynamic. We can just kind of turn on if it fails or not. Finally, I'll just kind of quickly go over the simulation examples. So the above domain model that I just introduced was then formalized using the approach that Jan Keur developed, and then put into a dedicated software environment in MATLAB. And then we could easily make these simulations with it, and you can kind of play with all the parameters. And as you saw in the model, we have all these like gaps between who is doing what and when. So it gets pretty complicated, and it's great to be able to like tune the parameters and see what works best for the model to run smoothly. So this is showing the world states. So the activation means that the state is actually happening. So for example, a call for intubation is starting on top. And then this kind of puts into motion all the other states in the model. So as you can see by this WS17 that is happening around timestamp 50, the verbalization of the success by the doctor is activated. So that means that this is a successful attempt. I'll quickly just continue to the doctor one. So we also have the nurse, but we're running a bit out of time. So as you can see for the doctor, the ownership states for the doctor, the dotted lines are being activated, meaning that the doctor is doing his or her tasks. And some of them are not being activated, and these are actually the failed attempt ones. So also here, the verbalization of the success by the doctor is being activated, which is this thick brown line. And then the nurse has very similar ones. I'm just gonna go over it quickly because our time is up. So just to quickly summarize, we made a computational network model for simulation of tracheal intubation. And in simulation experiments in the paper, both successful and failed attempts were addressed. Next steps will be multiple intubation attempts, allowing for adaptivity by the doctor and the nurse, and possibly having a bigger team size for this project. All right, thank you for listening.
So any questions from the audience? Okay, I have actually one question. As I can understand from the paper, it's this study was based on simulations. So what's your vision about the practical use of this system in actual hospital operating rooms?
That's a great question. Can you hear me well with the headphones?
Yeah.
Okay, awesome. It's definitely a challenge of like putting these simulation lines into practical use, but there's actually an exciting project being started at Technical University of Delft in the Netherlands, which is also going to use this model, hopefully. So you can think of using these kind of models to train doctors and nurses with simulations so they can go over the steps and see where possible mistakes in these shared mental models happen, so they can get used to communicate better or like improve their mental models. Or you can think of more like an artificial intelligence agent that really understands how the process is going and that can give real-time feedback while having certain sensors in the room or things like that. So yeah, that's kind of two directions you could go into, I would say.
Okay, thank you.
Thank you.
Any other questions? Okay, Professor Mohammed, if you would like to introduce the next presentation.
Okay, thank you. I would like to invite paper ID B219, title System Level Knowledge Representation for Cognition in Neuroscience, CSTRNS, Taiwan, China.
Okay, I'm going to share my screen for the next talk.
Good morning. I am presenting today system level knowledge representation for metacognition. The motivation for this work is to follow up on earlier presentations where I shared my challenges, the challenges that I encountered in mapping the biological neural networks knowledge representation to the artificial neural networks knowledge representation. So when I started to work in neuromorphic systems, I am not a neuroscientist, I'm a knowledge engineer. And when I started working at the design of neuromorphic systems, the first thing I looked for was, so how do we model the brain, the human brain, into systems? Where do we start from? And it has not been easy nor clear how these two models, how these biological and artificial neural networks match. And that's where the journey started into neuroscience learning for me. This was a few years ago, and I have presented my findings, which were mainly the challenges. And from there on, I started studying neuroscience as a whole, as a knowledge domain, because that's what I do. In my field, we look at knowledge analysis, and having addressed or having identified a number of challenges and open questions in relation to knowledge representation in neuroscience, I have then started addressing these questions in some research, and I'm presenting some of it today. So we should remember that learning is not just for students. Students have basic level learning, but researchers have to continue to learn. So these challenges the challenges that my research tackles apply equally to newcomers and new learners, as well as to experienced researchers. In fact, as senior neuroscientists may have knowledge that can become superseded by new knowledge, and therefore the learning process may even become steeper for experienced learners, for experienced practitioners. So learning is very much ongoing process and focus on motivation for my research. So not only my learning, but the design of information systems that are supposed to support learning. And then the ongoing learning process that goes in any advanced reasoning and any advanced cognitive process. So the focus, an important part of the background for this work is that we are learning about metacognition in complexity. So metacognition is the higher order cognitive processes, loosely defined as, which are necessary for learning and reasoning. So reasoning and learning and metacognition cannot be separated. It is necessary to include metacognition in any reasoning, especially in complex domains, and neuroscience has turned out to be one of the most complex knowledge domains I've been working with. And knowledge representation, which we normally use for communication and systems design as well as education, is needs to be stepped up to capture this metacognition. So here, the work is not just learning in terms of acquiring new knowledge for the purpose of advancing engineering. So here we are acquiring new knowledge in neuroscience that we apply in engineering. And a lot is coming out in terms of complexity, metacognition. So there is a lot of data out there in open data in relation to neuroscience. There is a lot of open data coming out every day. But when you try to use this data to, say, design your database or come up with a new processor design or a new system that models the human brain, this data is, as it is useless to most people. So unless you have worked with this data and you understand it, or you understand the software it has been used to put it together, or unless you have understanding and knowledge of specific software skills, this data doesn't really say much. And acquiring the knowledge to query this data is it's a massive task. So to make this knowledge, this data useful, it is necessary to present it with meta-knowledge. It is necessary to identify and express system level. So what this paper is about, in essence, is identifying the system level and representing in such a way that the existing data can be fit into, say, metacognitive model that can be used for learning and reasoning. So here we have, I just like to remind that the challenges faced in carrying out this task there were a lot of data models in use, fragmented. There is a very vast landscape, and that these landscape can only be addressed with meta-representation. However, we don't have meta-representation yet. This research takes a step in that direction. And we have to keep in mind that there are trends such as systems neuroscience emerging. Again, system neuroscience, very vast. Meta-science is happening. At the back, where the scientific paradigm is also evolving to become more integrated. And we are understanding that metacognition is central to our learning and reasoning. So basically, fundamentally, the paper shows the knowledge representation object, which is here shown as a diagram, a relational structure, a grid. New research confirms that grids so relational structures are used by human cognition and metacognition for the processing of complex knowledge. So this knowledge object is symbolic, so to speak, is an example. And it attempts to capture a number of factors which play a role in our processing of neuroscience data. So when we get a construct which is embedded in the meaning of which is embedded in a term, which in turn represents a concept, this knowledge to be meaningful and to be better processed in the absence of any other fact or limited number of facts it needs a number of things. So we have a concept in the context of metacognition, which needs to be represented using a higher level, higher dimension, higher abstraction, which we call meta-representation, following one or more processes, fulfilling a function, and being part of a system. So the knowledge representation object attempts to identify the units of knowledge, so to speak, which could be data, any datum, and related to the language, the way it is expressed, and to the process, the inference and reasoning process, which is part then, and that supports a decision, which in turn informs action and prompts a behavior. So taking a behavior decision, structure behavior function, onto a behavior decision action, say grid, we get some support into sorting out, processing this neuroscience concept and term. This model, the knowledge representation object, can be read in conjunction with the method proposed in the other paper, system level knowledge analysis. So the other paper tackles a similar problem in relation to learning more specifically, and it aims at putting together a number of elements and factors that contribute to that constitute the knowledge, complex knowledge, applied to obviously the neuroscience domain. And what the whole thing is aiming at is making the case for a system level knowledge representation. here an object is designed and proposed. The object can be used to navigate and support the learning manual. So just by reading a book or reading books, taking resources and mapping the data onto a grid, it will help us create the metacognitive structure. But also, and more importantly, I think, it aims at preparing for engineering design of neuroscience educational systems, as well as automation of future systems that adopt brain data as the architecture, the basis for their architecture. So thank you very much. Please get in touch if you have any questions.
Thank you very much for your nice presentation. Now I ask the audience if you have any question. So I have a question that is in your model, how you will ensure your knowledge representation schema that you have presented with the brain interface.
Thank you. Sorry, can you repeat the question?
Can you please link your knowledge representation schema that you have presented with the brain interface, human brain interface?
Sure, that's a very long answer, but let me give you a short answer. It depends what the brain interface is supposed to do. And this is part of the system design problem. Depending on what is the end system supposed to be doing. But this level, which is fairly this is the most abstract level, and there will be follow-ups and drilling down these areas into more specific tasks, but it's ultimately trying to place the knowledge from the knowledge base, any knowledge base. I have worked, for examples, with books or even with the conference proceedings. Take any conference proceeding like this conference and try to map the concept to into a system and say, what do I do with this knowledge and how do I design a system from using it? Okay, this is the most basic example. And so depending if the system is supposed to perform some cognitive function, it will direct or model the knowledge accordingly. But at some point we expect, you see, this is a question which came up a little bit from the keynote, one of the keynotes yesterday. So all this histological data that we have, all this cellular level data, we don't have ways of mapping that to the behavior function level. So we still, in general, I'm speaking in general, when you study the neuroscience corpus as a whole, either from books or from conference proceedings, you don't find how does this fundamental physiological structure of the brain impacts or reflects into the behavioral and cognitive. And one of the reasons I'm now learning why this is not happening is because a lot of the data is not from humans. So the data which exists in neuroscience is doesn't have the function, doesn't display eventually, doesn't belong to physiology or brains that have the functions that we are trying to model in the system. Or maybe only parts. So, I mean, vision or, smell, it may have, but I'm talking about higher level cognitive functions. So what the model does, or it hopes to do, and this is a beginning for this, and I hope people will help me build it. Like, please do criticize it. Please do crash tested and let me know if it works or not. But ultimately, what the model wants to do is saying we need to start processing all this amount of neuroscience data which is coming out from research and using it to develop systems. But at the moment, it's because it's so much and because it's not particularly clearly labeled, not at the level that we need system design level, then it's very much work. So it's an encouragement also for the data producers, whoever is producing brain data, to please start labeling the data in a way that the people who want to use it for some system design know what it is and know whether it can be useful to them. And this eventually, when this entire process is going to be automated, it will be designed into interfaces, which you in your question referred to as brain interfaces, but eventually they're going to be mind-computer interfaces, which is what we are aiming at. Have I answered your question?
Yes, thank you very much.
Thank you for the question.
If the audience has any question? So if not, we can proceed to the next paper.
Okay, so the next paper is numbered BE226. Title of the paper is Brain Connectivity-Based Classification of Meditation Expertise. The authors are Pankaj Pandey, Pragati Gupta, Krishna Prasad Miyapuram. I'm going to share my screen right now. The paper will be presented by Pankaj Pandey.
Hi all, I am Pankaj Pandey. I am PhD student at Indian Institute of Technology, Gandhinagar, India. Today I present our study on brain connectivity based classification of meditation expertise. The objective of this study was to learn patterns from electroencephalography data. Here's the EEG pipeline starting from data acquisition, followed by the pre-processing steps to create features, and lastly train and classify the model in different conditions using different measures. Now I will discuss pattern learning in brain signals captured while practicing meditation. But before that, let's understand meditations types. Various types of meditation capture different aspects of brain states. It elicits common and specific neurophysical and logical changes in the brain, as well as activating different areas of cognitive ability, such as attention, metacognitive awareness, cognitive control, compassion, etc. Such as here, we can observe 4 broad types of meditation practice, which encompasses focus attention, open monitoring, transcendental, and loving kindness meditation. Each has its unique style of practice and sometimes an interlink with various other meditation traditions, such as in focus meditation, selective attention to a particular object, such as breathe, visual image, etc. Whereas in open monitoring meditation, the aim is to remain attentive to any experience that may arise without focusing on a particular object. And transcendental meditation, the goal is to use a sound or a mantra to be aware of the present without an object of thought. Loving kindness is to nurture love and compassion toward oneself as well as other beings. Similarly, in some of the meditation types in focus attention includes Himalaya Yoga, mantra metta meditation. For open monitoring, Vipassana, Isha Shunya, and Zen. So now we highlight a few of the recent papers of various meditation tradition using different methodologies to study EEG oscillator activity, such as I start with the first one, utilizing time frequency analysis to study EEG correlates of experienced meditators, where they observe increased frontal, midline, theta, and somatosensory alpha among experienced meditators. Another study highlights three groups of meditation tradition: Himalaya Yoga, Vipassana, and Isha Shunya, utilizing EEG through spectral analysis. They observe higher gamma amplitudes for all the 3 groups of meditators, and especially alpha activity for Vipassana meditators, and lower 10 to 11 Hz alpha in Himalaya group. In the recent study, individuals practicing 3 types of meditation: tradition, focus, attention, loving kindness, open monitoring. In this, result indicated distributed delta networks, left hemispheric theta network, and right hemispheric alpha network for all the 3 types of meditation techniques. As even one type of meditation practice has various aspects in it, and it differs from individual to individual. Here, in this particular study, we utilize publicly available data set on Himalaya Yoga meditation, which includes 12 experienced meditators and 12 non-experienced meditators. So here the style of practicing meditation in Himalaya, mostly mentally reciting the mantra. And when deeper level meditation or stillness of the mantra repetition gradually cease. This is the sequence of experimental paradigm. Experience sampling probes were presented at random interval ranging from 30 to 90 seconds, and after each experience sampling probe series consisted of 3 questions. The first question: please rate the depth of your meditation, for which participant evaluated the subjective depth of their meditative state for the moments immediately preceding the first probe on a scale from 10 to 13 by pressing corresponding key on the keyboard. After their response was registered, similarly the second question: please rate the depth of your mind wandering, and followed by please rate how tired you are. The time range of the experiment lasted from 45 minutes to 1 hour 30 minutes. Our analysis based on the 20-second epoch trial ranging from 20 seconds prior beginning to the question 1. Here I explain complete pipeline from acquisition to classification between expert and non-expert meditator. The first stage is to data acquisition, which is followed by second stage epoch extraction. In practice, varying the number of epochs has been suggested to obtain the best set for computing the connectivity matrix. We utilize 20-second trials in 2, 4, and 8 groups. Signals were filtered using band pass to obtain the frequency band of interest, all the frequency band like delta, theta, alpha, beta, and gamma. To obtain the significance of regions, the signals were classified based on the few scalp regions and their combinations. Phase locking value between the 2 signals was generated across time points. MPL matrix were converted into a vector, including correlated variable. These 3 thresholds were used to remove correlated variables and to create a feature vector. Last stage is to train models. We use 5 different models with hyperparameter tuning so that the variability of the data would be covered. We use 3 validation techniques to report the accuracy. These are leave one out subject, leave one out session, and tenfold validation. This illustrates the 5 primary scalp region based on the electrode placement, different positions, and the table defines these 4 combinations. And this one is to process to compute phase locking value between 2 electrodes. Hilbert transform was used to calculate the instantaneous phase value, which was further used to compute the phase locking value. The results we have, we achieved a maximum classification accuracy of 91% in loose validation, and in loose and tenfold in 2 epochs, and 80% in loose in 8 epochs. Means here 90% in this loose and 80% around the maximum accuracy in loose. So all 3 validation techniques perform above 80% accuracy in 8 epochs. Theta band showed maximum accuracy, followed by the delta band. Classification accuracy of 91% was achieved in delta and theta, whereas alpha, beta, and gamma obtained 84, 89, and 86 respectively. To observe each band varied in their performance across region. The top box in the figure represents central, parietal, and their combination. The middle box indicates frontal and combination of frontal and central, and the bottom box includes 2 frontal, central, and parietal combination. The most dominant bands across central and parietal regions were beta and gamma. We found the most discriminating characteristics from the frontal, comprising theta and alpha bands, with we with few features, and with few features here in the correlation, with few features the model can learn efficiently and become more interpretable and less biased. We removed the correlative variable and observed the highest perform with correlation threshold of 80. QDA and Gaussian process classifier outperform other mentioned classifiers. So here the results we observe beta and gamma oscillation dominant over parietal regions, which is a marker attentive globe state, and alpha and theta oscillation dominant over frontal region. That's a attentional state of awareness. Here we represent like frontal region, central region significant discriminator with accuracy greater than 80% across 3 validation technique, and like FFCC in these combinations showed accuracy greater than 90% tenfold leave-one observation. Means when we include all these FFC and PCP means all electrodes, you can say. And the third part, a quantitative discriminant analysis Gaussian process outperform other classifiers and highest accuracy in the tenfold and leave-one out session. Here I discuss some future applications. As we know, digital content and gadgets are extensively available with significant advancement in digital technology and system. As a result, there has been an increase in information overload, possibly leading to overuse of the sensory, perceptual, and cognitive circuits involved in the process. Neurofeedback aided meditation may lessen the attention grabbing power of mind wandering and spontaneous thought processes both during practice and in daily life, which may ultimately assist in deepening meditation practice. Due to several benefits of meditation, attention management, well-being, states of flow. So using the state-of-the-art deep learning and machine learning model, this can be achieved to provide real-time neurofeedback among named practitioners. So this is the model like in the neurofeedback domain, where it can be seen as a signal acquisition and processed by pre-processing step and just feedback to using application interface to the participant. So this is one of the good application in using these learning from expert meditators and can be transformed into a digital portable devices. So these are the future application and these are the references, and thank you.
Okay, any questions from the audience?
Yes, I have one, if I may.
Please go ahead.
Thank you for the talk. And so I'm interested to learn, if you know, these patterns that you summarized in your one of the last diagrams with all the boxes and the colors of the bands, these patterns of waves or frequencies, in your knowledge or best guess, do they match the any other activity? So can you see these patterns in other activities other than meditation, like I don't know, sleeping or I don't know, whatever? So are they unique to meditation, these patterns?
Yeah, this is a very good question. But in some, like in sleep, delta is more important. But in theta is also in relaxation, like in relaxation, alpha and theta are more important. But you are asking for a unique signature, like whether this is a unique signature or not. So actually it is varying across different meditation tradition. So like the subject, what it is experiencing, is it related with the same signature? Like there are two different meditators if the signatures are same, but are they experiencing the same thing? So it's a current is ongoing study. It's only people are just finding the neural signature, and similarities is only with the state, relaxation state or something. But there is a band, if we say power, in some traditions, there is a same frequency band. You can say that there is a high frequency band in like in gamma. There is a high frequency band in experienced meditator. So this kind of signature currently we are able to see.
Thank you very much. Yes, I would be interested in seeing the method, I mean, more data, and then testing various hypotheses.
Okay, sure. Thank you.
Any other questions? Okay, Professor Muhammad, please present the next talk.
So I think we can ask our today's last topic, paper ID B241, exploring the brain information processing mechanisms from functional connectivity to translation applications. The authors are Moonji Koen, Meiji Institute of Technology, Japan; Jianhu Chen, Beijing University of Technology, China; Jiokui Tao, University of Southern Queensland, Australia; Azuyuki Imamura, Mebashi Institute of Technology, Japan; Ipen Liang, Capital Normal University, China; Lin Jun, Mebashi Institute of Technology, Japan. So we can now play the presentation.
Hello everyone, I'm so honored to be with you to communicate academic questions and ideas. The title of my presentation is Exploring Brain Information Processing Mechanisms from Functional Connectivity to Translational Applications. So the content is listed as follows. In this paper, we focus two key questions. So the one thing, we focus on a basic problem about how brain function get rise to mental activity. Another thing, we focus on an extended problem about translational study. More specifically, we use the functional MRI technique and graph topological methods to decode brain data. It has become one of the most important strategies to explore brain information processing mechanisms. First, let's think about the most common brain research process. In this process, an initial hypothesis must first be given, then a single experiment is designed to collect brain data, and the relevant analysis methods are executed. Obviously, this strategy is essential to understand one-to-one relationship between brain structure and cognitive function from a single perspective. But when we consider the interaction between several cognitive functions, this strategy is not enough. So, we need to change our view from single to systems. More specifically, under a brain investigation process, we hope handle multiple brain data with different hypotheses continuously. We know from brain data it involves complex mental processes, complex experimental design, complex data states, and complex analysis pipelines, and tens of thousands of brain data heads constructed as big data. The key challenge is when we want to investigate a complex brain science problem, how to select and fuse brain data from the data ocean to realize systematic computing. Facing such challenges, we learned the systematic methodology about brain informatics, It treats the human brain as an information processing system and leverages the benefits of cognitive neuroscience, big data, and artificial intelligence to realize the systematic, integrative, intelligent, and multi-aspect investigations. Following this methodology, this paper proposed a systematic approach. It mainly includes the conceptual data brain model and never-ending learning. Then, we presented 2 case studies about functional connectivity and translational applications. First, we designed a conceptual data brain model to manage complex brain big data. In this model, we design the concepts of brain function, cognitive experiment, brain data, and analytical methods. We also design their relationships. Based on the conceptual data brain model, we constructed a sample library. We can find these factors correspond to concepts from the conceptual data brain model. Then, we consider how to select the brain data we want from the sample library. Here, we propose a systematic experiment design modeler. It includes 2 types of experiments, namely the main experiments and supplementary experiments. As their starting point, we first design a main experiment by matching these 3 factors from the sample library, and then we compare these 3 factors between their main experiment and their samples from the library, and we select the most similar sample as the supplementary experiment. These selected brain data are analyzed by the functional connectivity methods. More specifically, we use AAL brain template to construct brain networks and compute their degree centrality and NG vector centrality at various densities. Finally, we determine the significance of each node by using statistical methods. And these methods are treated as evidence to input the next modeler. This page tells us how to fuse the conceptual computed results. First, we need to infer the essential types and their weights, lambda, for the computed results. For example, in the left finger, the VM indicates the functional domain with respect to the main experiment, and the VS indicates the functional domain with respect to the supplementary experiment. These functional domains are concepts from the conceptual data brain model. So, if their relationship is recognized as descendant, the selected evidence belongs to the type 1, and its weight equals 1. At the same time, if their relationship is recognized as sampling, the selected evidence belongs to type 2, and its weight equals -1. The last, if we get the evidence and their weights lambda, we can fuse this evidence into a tau value. Here, the lambda is from the evidential inference. The mask indicates the evidential overlapping, and the R indicates the computed results. Finally, we can integrate all modelers as a loop to realize never-ending learning. Next, I will present 2 case studies to further interpret the proposed method. Here, we take the human reasoning understanding as an example. So, in the first loop, the D7 is selected as the main experiment because the data optioned by a human reasoning task. Then, the D1, D5, and D2 samples are selected as the supplementary experiments, one by one. Their degree centrality values are computed and fused to realize never-ending learning. This page shows the results measured by ng-vector centrality. For more details, this page lists some brain regions selected by the never-ending learning process. This table shows that these brain regions are related to human reasoning, especially in the frontal lobe. In this paper, we don't make a conclusion about which brain region is exclusive for a specific cognitive function. But what we want to emphasize is the importance of changes along with never-ending learning, because we can find that the tiny tweaks can lead to big changes. Now we think about, is their results meaningful and useful? So we performed another case study. We consider the translational application problems. The basic hypothesis is some cognitive impairments may be more easily arise in a mental disorder. Here, we selected the D6 dataset from the sample library. This dataset includes the resting state fMRI data and the affective picture-induced fMRI data from 19 patients with major depressive disorder and 19 healthy controls. The right figure shows the experimental details. This page shows the details about translational applications. These feature patterns are from the selected nodes in the AAL brain template. We selected the first 12 nodes with bigger tau values, which have bigger specificity for the emotional phase matching task. Here, each 3 nodes are treated as one pattern, and the classification model is constructed by support vector machine algorithm. This page shows the classification results. First, using these feature patterns, we can find it can achieve around 95% accuracy, whether resting state or task state, especially from L5 to L10. And these results look like stability. In addition, these results also give us inspiration for understanding relationships between brain function and mental disorders. For example, in this case, the large fluctuations arising from L1 to L5, it indicates that there may be not the emotional phase matching impairment in the current MDD population. It is summary and feature research. In this paper, we focus on the open question about complex function structure relationships. We highlight that the single data analysis is necessary, but it's not enough. So the statistical methods is becoming an urgent need. In this paper, we introduce a data-brain-driven statistical approach to explore the brain mechanism from functional connectivity to translational applications, and 2 case studies were performed. In the future, we think about the brain big data is still one of the most important directions, which can be handled by the conceptual data-brain model. In addition, brain science also welcomes open science to improve scientific cooperation. By meeting big data and open science, we can enrich the data-brain model to realize more translational applications. That's all. Thank you for your attention.
Thank you very much for the nice presentation. Now I ask the audience, if you have any question, please feel free to ask the speaker.
Yeah, I have one maybe short question. So in the results, you had a figure where you're showing the accuracy versus the levels L1 to L10. Can you explain more what those levels actually represent?
Okay. In this presentation, thank you for in this presentation, we first we select the feature patterns through the functional connectivity analysis. So we use our proposed model to measure the different nodes from in the automated anatomical atlas. So we measure the about 90 nodes with different weights, and this weights indicates the importance for different nodes. So we select the first 12 nodes with bigger importance values. So this means that these nodes have bigger specificity for specific experimental task. So for those 12 nodes, we make each 3 nodes as one feature pattern. So we can combine this feature pattern and the classification models to realize detection for patients with major depressive disorder and health controls. So this is my point.
Okay. Thank you so much. So this concludes the session, the track 1. I would like to thank all of the authors and presenters for the nice presentations. And Professor Mohammed, if you have any concluding remarks.
I think we have seen a very nice presentation and thoughtful research from the research side. I think that's all from me.
Thank you so much, Professor Mahmoud.
Yeah, well, thank you very much for chairing the session. Now we are almost at the end, in the sense that in like a few minutes we'll start with the closing ceremony of the conference. And as there are 5 parallel sessions, some of the sessions are running slightly late, and that is why I would request you all to be a little bit patient. We were supposed to start it at 11:30, but taking that into consideration, we decided to start it at 11:45, which means that delay the session by 15 minutes so that, well, the other sessions can also end. And especially, well, all those session chairs who have done amazing and fantastic amount of work since last 2.5 hours also should probably get a cup of coffee or tea and then relax themselves a little bit. So from that particular perspective, well, we will start the ceremony, the closing ceremony of the conference in like 10 minutes time. So there is a 10 minutes of refreshment break. Yes, please.
Did you see the Jing already in the room?
No, I don't see Zing here in the room. No.
Maybe we should wait her to join. All right.
No, she's joining now. Yeah.
Yeah. Okay, let's start this conference closing session. Welcome to join this closing session of the Brain Informatics 2021. And my name is Ning Zhong. I chair this closing session. So I believe everyone enjoying this conference high quality technical program. There are excellent keynote talks, high quality paper presentations, and workshops, special sessions that focus on the different hot topics. At first, I would like to invite Mufti to give the summary of this year's conference. Okay, please, Mufti.
Thank you. So just a second that I will share my screen. So as for the summary of the conference, well, thank you very much for attending all these 3 vibrant days that we had. So these days were quite busy. Okay, so day 1 we had 4 keynotes, 7 workshops, and 2 special sessions. Day 2 we had again 4 keynotes, 3 workshops, 1 special session, and day 3 today we had 1 workshop and 5 parallel technical sessions. Altogether, we had 8 keynotes, 11 workshops, 3 sessions, where 50 contributed papers were being presented and 18 abstracts were being presented. So a little bit about the submission and the peer review process. So we received altogether 90 submissions this year. So each of the submissions were reviewed by at least 2 independent reviewers and 1 program chair. Finally, we had accepted 50 papers, out of which there were 48 full papers and 2 short papers. I'm very glad that the proceedings of the conference is online, already, and we could actually get the accepted abstracts also as the preface included in the proceedings itself. So all these abstracts which were accepted in the conference also are present in the proceedings, in addition to the all full and short papers. So well, the access to the conference is complimentary, and it would last for access to the proceedings is complimentary for 4 weeks, starting from 16th, that is 1 day before the conference. So please, I would request you to access these proceedings through the conference website and keep or download your personal copy of the proceedings for future reference. And well, during these 3 days, we had 18 session chairs. Okay, so 10 session chairs running the parallel sessions today, and 8 excellent keynote talks that we had, the chairs who had chaired those keynote sessions. Additionally, there were altogether around 25 workshop and special session chairs who have contributed towards the success of this conference, without whose unconditional support and assistance this conference would not have been possible. So I would like to say a big thank you from the organizational perspective, that while the technical program that we have presented here has been very heavily supported by you people, BI 2020 Technical Committee is really indebted to you. Thank you very much. Back to Ning Zhong.
Okay, thanks, Mufti. So I also mentioned this. Really, we received many excellent papers. Yeah, also give the excellent presentations. So we will have the virtual best paper award ceremony. I would like to invite Stefano to chair this best paper award ceremony to announce the awards. Please, Stefano.
Thank you, Professor Tsong. So let me thank you, everybody, again, and for their enormous effort for the organization committee and the people who chair the sessions. I know it was a great effort, and let's hope that next year this will be done in person. So it is the moment to give the awards, and let me share my screen then, starting the virtual award ceremony. So I hope you see it. Okay, so the first award is the best student paper. So basically, the winning paper is titled "Exploring the Brain Information Processing Mechanisms from Functional Connectivity through Translational Applications." And so the authors are Hunzi Kwai and others. And so I really congratulate with the authors for this achievement. So congratulations. So the next award concerns the best application paper. And the title of the winning paper is Development of an Artifact Removal Module for Extracellular Neuronal Signals in a MATLAB-based open source toolbox. The authors are Marcos Fabiezi and others. And again, I really congratulate for the important achievement. Then we have the final award, it's the best paper award of the conference. And the best paper of the conference, and this goes to the paper titled Spectral Properties of the Local Field Potentials and Electroencephalograms as Indices for Changes in Neural Circuit Parameters. And the authors, I hope you hear me, the authors are Pablo Martinez, Canada, and Stefano Panzeri. So really congratulate with the authors again for this achievement. And thank you all for putting your effort, your words in this conference, and letting us appreciate your recent achievements. There are, of course, corresponding certificates that are given virtually. So let me go through them. So this is the best student paper award. I think, Mufti, there was a little mistake here. I had briefly corrected it. Please revise it. And then send the certificate to
Yeah.
The awarded people. And then this is the certificate for the best application paper. And this is the certificate for the best paper award. So let me stop sharing, and I give back the floor to Professor Tsong for the concluding the conference.
Thank you, Stefano. So, as you know, our this year conference originally we planned to organize this year conference in Padova, Italy, but due to the COVID-19 pandemic, we finally this conference be converted to the full virtual. Yeah, but I think everyone also want to visit Padova. Yeah, we want to meet each other physically, in person, not just virtual. Yeah, but so I hope we also plan the next year conference. Yeah, so I mean will be held in the Padova, Italy. But we also another good proposal from the University of Queensland, Australia. Then finally, we decide next year conference should be co-hosted by University of Padova, Italy, and also University Queensland, Australia. So at first, I would like to invite Stefano to give the more information, introduction of the Padova, also University of Padova. Please.
Okay, so I think Mufti should have at least a nice video to share, a pre-recorded video, of course, concerning the beauties of Padua and the importance of the University of Padua, so that people knows what can expect from the next year visit.
Okay, so this is essentially a video which is quite popular regarding Padua. So this is not something which is prepared by us, but this is something which is shared by a very popular explorer. So let's have a look at that video.
Probably should the full screen. Go to the full screen.
The night is not yet over, but Padua is already awake. And today, like every day, the life of the city is reflected in its university, a home to free men and women and to freedom of thought. This is where figures like Copernicus, Andrea Vesaliu, William Harvey, and Giovan Battista Morgagni formulated thoughts that have changed the world. This is where modern medicine began, taught for the first time at the patient's bedside. This is where Galileo invented scientific method. The University of Padua looks back on 8 centuries of history, but it also looks forward to an exciting future. Palazzo del Bo throws open the doors to its courtyards and its 15th century anatomy theatre. The University Botanical Gardens, the oldest in the world, welcome their first visitors of the day, each eager to admire the wonders inside. In Padua, the new day has finally begun. The old and the new encounter each other once again. And from this encounter comes constant stimulus for innovation. Here is the lifeblood of Padua's thriving industry and commerce, the driving force of an area that derives its energy from its university. On foot and by bike, students mold the very geography of this city. In their thousands, they are the lifeblood that connects every road and street in Padua to the lecture halls, libraries, and laboratories of the university. Their faces and voices are among the first signs of the city astir. Inside the university, another day's work is beginning. An extensive, if sometimes invisible, workforce is ready to support every activity. New projects need to be started. New investments need to be made. A highly complex set of services has to be provided. The research laboratories are already acquiring new data. Results and experiments are being evaluated and debated. From the earliest manuscripts to tomorrow's technologies, research at Padua covers every aspect of scientific inquiry. And as always, what drives this research is human curiosity, that thirst for understanding which the university nurtures in every student, in every researcher, in every member of staff. When the morning is over, the university seems to empty. Its population emerges onto the streets of the city. People take time out to grab a bite to eat and have a chat, to meet friends, and perhaps make new ones. It's a moment of relaxation snatched from the day's busy timetable. Then it's back to work. The schedule for the afternoon is varied. There are more lessons, time for some last-minute revision, and somebody in the next room is already discussing their degree thesis. Somebody is standing in front of the Great Hall. This is where the university welcomes its most illustrious men of learning. The university is also a focal point for leisure activities. You can pursue your interest in sport, nurture your dreams of theatre and cinema, and there's still time for yourself and for others. Then evening comes, sounding the last notes of an intense day. Now it's time for getting together, for that unforgettable student social life. On nights out, the city's meeting places play a starring role, or you might prefer to relax at home in one of Padua's many halls of residence or student digs. Padua seems to be sleeping again. But on a crystal clear night in Asiago, high in the hills, the domed roof of the university observatory slowly slides open. The focus is on the stars and the depths of space. The night reigns, but the university never sleeps.
Okay, thank you very much, Mufti, for sharing this video. So as you have seen, there are many things to visit in the university and also Padua. So I really hope that next year we will be all here in person, and we will shake hands, and we will have basically a normal behaving life, scientific life together during this conference. Of course, the next year conference is as Professor Tsong clearly anticipated, there is a partnership with the Queensland University. Will be co-hosted basically, so physically in Padua, but also Queensland University will be paired to the University of Padua. So it is my honor to ask Jin He to introduce also the Queensland University. Please.
Thank you for the this year's wonderful hosting for the conference, Brain Informatics Conference. It's such a good experience for me. And thank you. It's a very great honor for me can present next year. Basically, due to the pandemic, it's uncertain somehow if we can travel to Europe or not next year. So we propose to host something like the Twin Cities conference for BI 2022, and all of the Asia Pacific participants can get the chance to attend the conference while visiting the University of Queensland. So I would like to share my screen. Maybe the host has to give me the right.
Yes, Jing, you have it. Yeah. Please go ahead, please. Yeah.
you see my screen now?
Yes.
Yeah. Yeah, thank you. And maybe you guys will be curious what is the University of Queensland and where is it? Basically, that's a big sunshine city and the Sunshine University as well. I just you just use the Google to search the University of Queensland and you will find that this is today's website of the University of Queensland. You might say it's quite purple. Yes, we do have very beautiful flowers, around the campus. And you guys might, got the chance to visit Australia. So I would like to share 3 videos. The first one is going to introduce Brisbane. Actually, Brisbane will host the Olympic Games for 2032. And the second video is going to introduce the University of Queensland because our brain informatics thing. We are going to host our offline conference in the Faculty of Medicine, Capitol Lane. So I would like to share 3 videos. Maybe I think, I'm not quite sure if I can come to the video right now. So try, I might share again.
The capital of the state of Queensland, Brisbane, is located inland from Moreton Bay, between the Sunshine Coast and the Gold Coast. The heart of the
You need to
hilly
share it. We can hear it, but we cannot see it.
to the Coral Sea. Queensland is Australia's sunshine state and enjoys a subtropical climate with hot, humid sun.
Sorry, technical problem of Euro.
Yeah, in this case, maybe just share the slides, not video. Yeah, good.
Yes, now we can see that. Yeah.
Yeah.
The capital of the state of Queensland, Brisbane, is located inland from Moreton Bay, between the Sunshine Coast and the Gold Coast. The heart of the hilly city is its river, which snakes from Mount Stanley to the Coral Sea. Queensland is Australia's sunshine state and enjoys a subtropical climate with hot, humid summers and warm, sunny winters. In Brisbane, any time of year is a good time for adventurous outdoor activities along the river or coast. Experience the more relaxing side of Brisbane in the parklands, museums and bars. Brisbane still has the laid-back country feel that is so iconic for the land down under. However, it's Australia's 3rd largest city, houses over 2 million people and is growing fast. Start your tour of the city on Brisbane's biggest hill. Mount Coot-tha was already visited by the Aboriginal people up to 40,000 years ago, and its name means honey, which they found here in abundance. While you're here, explore the wonders of the Southern Hemisphere in the Planetarium and Brisbane Botanical Gardens, including the Laughing Kookaburra. Oi, what are you laughing at? Feed lorikeets and see iconic Australian wildlife at the nearby Lone Pine Koala Sanctuary. Meet kangaroos, koalas and wombats, and don't miss the animal shows, which demonstrate some real Aussie traditions. Downriver, in the central business district, the Queen Street Mall displays all the country's big retail brands. Visit the nearby Brisbane Museum to learn about the mining boom and other events that shape this city. Exhibits show how World Expo 88 led to the transformation of the river's southern bank, the city's favourite backyard, which even has its own beach. Spacious South Bank has many free activities and facilities which attract more than 10 million people every year. Cool off in a lagoon or browse the weekend markets. Join the locals for a beer or visit one of the many prominent museums and theatres. The Wheel of Brisbane overlooks the Cultural Forecourt, a collection of Queensland's major museums and theatres. Meet some prehistoric creatures, admire Indigenous and European modern art, or see a show at the Performing Arts Centre. From the piers at South Bank, take the public ferry downstream to New Farm Park, where the restored powerhouse has become a real institution among art and music lovers. On your way back, hop off at Kangaroo Point to climb the Story Bridge, one of the best spots to watch the sunset over the city. Book your guided tour for mid-afternoon because nightfall comes early in Queensland. Get an early start and jump on the ferry to Moreton Island for a fun day out at the beach. Join a Segway tour along the shore before surfing down a massive sand dune. On a hot day, snorkel around the Tangalooma Wrecks, or if you'd rather stay high and dry, book a helicopter ride instead. Flying over the bay and the Coral Sea, it's clear that Queensland's natural beauty merely starts in Brisbane. 2 hours flight to the north, the Great Barrier Reef is a living national treasure. Brisbane has a wealth of attractions, and you can indulge as much as you like. But in the river city of the north, you're never far from nature, which is free for all to enjoy.
There is some noise. Yes, there is a lot of background, so we cannot really hear you speaking.
Yeah, that's okay.
You can't hear me what I'm talking about?
Yeah.
Now somewhat okay. Yeah.
Not here. No.
The future has potential most of us can't even imagine, particularly in medicine. From new technologies for preventing, diagnosing, and treating disease, to revolutionary discoveries in everything from public health to neurobiology. The research we do has the potential to transform millions of lives. Working with patients from day one, the smallest interactions can make the biggest difference. From the Australian outback to the other side of the world, wherever medical science leads us, I want to be there. I need visionary, dedicated, and inspiring leaders, access to cutting-edge facilities, and support from like-minded, passionate peers. Together, we'll question everything, go further, and reach higher. I'm saying yes to the future, wherever it takes us. The University of Queensland, own the unknown.
We cannot hear you, Jing.
Yeah.
Okay, pass on to you.
Back to Bhupti.
Yes, back to the chair, please. You can't hear me.
Yeah. Yeah, maybe Jing said back to the Bhupti to give the more detail of next year program.
Okay. Yeah.
Organization. Yeah.
So allow me to share my screen. So I hope all of you can see. So I'm going to start with, again, probably many of you have seen this video at the beginning, but well, it is important at this stage also to reiterate a little bit about the history of this Brain Informatics Conference. So I'm going to run this brief video of 4 minutes. Thank you very much. Now let me move to the real organizational stuff. Okay, so again, as you already have heard and you were aware that we have been trying to hold this conference in Padua for a number of years now, but well, as we decided to hold this in Padua, the pandemic just disrupted these last 2 years. So this is the final effort in view of the University of Padua's 800th anniversary. So as we all know that well, Padua University was established in 1222, and well, this 2022 is going to mark the 800th anniversary of the University of Padua, and in view of that, well, we are going to give it another try, and hopefully well, we will be able to have it this time during the 15th and 17th July 2022 at Padua, Italy. So the theme is going to be very similar to how it was this year: innovative informatics for innovative treatment of brain disorders. And taking that into focus, as we already have heard, that it is going to be an hybrid event co-hosted at Padua, Italy for the in-person side, and at Queensland, Australia, the online side. So for the in-person ones, okay, so I'm going to provide some more detail. What are the specific things that made us to choose Padua? How can you reach to the conference venue, and all those specific details a little bit. Okay. So as you know that Padua is a city which is very close to Venice, around 35 kilometers, and there are a number of different nearby cities. For example, Verona, the house of Romeo and Juliet. Okay, so there is the beautiful Florence, we all know. Then there is Milan, Genoa, Rome, whatnot. Everything is within, let's say, 3 to 4 hours train journey. Okay, maximum Rome is like 4 hours train journey by the fastest trains, and Venice is like half an hour by train. So that if you are traveling by train, the Venice being the last stop of the train journey, Padua is just the station before, which means that it is just a 30 minutes, actually to be precise 26 minutes train ride from Padua to Venice. So well, it makes it excellent place for the conference, in addition to the technical excellence and the scientific excellence that you have seen at the University of Padua. And also there is yet another reason that next year actually in Padua there will be the WCCI, World Congress in Computational Intelligence, which is scheduled to be held from 18th to 23rd of July. So we will have just 3 days before that event. So our event will finish, the brain informatics will finish, then the WCCI will start. So well, Padua is also a city which is very well connected, it is very well accessible. So there are 2 international airports within, let's say that one is within the 30 minutes and the other one is 2.5 hours by train. And then there are many direct international routes, for example, like Emirates flights from Dubai and connections to Asia, Australia, and Australasia. Everywhere there are plenty of connections in there. And of course there are, for the European travelers, a lot of options. For example, there are 5 very neighboring airports, local airports, European ones. So one is in Verona, Treviso, Bologna, Trieste, and Bergamo. So they serve to all major cities and over 400 daily flights. So plenty of them. Well, now we are talking about the normal times. We hope that things will get back to normal by next year, by the organization of this conference. So there are almost 400 daily flights, and of course over 30 daily direct trains from Milan and Rome. So that is something which makes Padua to be very accessible. And of course, well, during the July, though it is going to be a little bit hot, but the days are really long. So there are these specific the squares and piazzas where people can just relax and have their dine in, have an evening cycle, and well, enjoy their long evenings as well. So, me being also an alumni of University of Padua, I really feel nostalgic well, I cannot share enough, let's say, the things that University of Padua has got to offer. In addition to the videos that we have seen earlier shared by Stefano, there is also yet another brief video that I also would like to add here. Please join in and be patient with me with this.
Padua, a beautiful and ancient city in the north of Italy, in the region of Veneto. On the one side of the city is Venice, which lies half an hour away. On the other, 10 minutes away, is Monte Grotto, famous since Roman times as a spa resort in the beautiful green Euganean hills. The Bacchione River winds its way through scenic quarters of Padua. Its banks offer great pastime possibilities for everybody. The perfume of history lingers in the heart of the city. Students from all over the world have fallen in love with it for centuries. As one of the oldest university cities in the world, it offers a vibrant atmosphere with charm that is irresistible, similar to the Prato della Valle. In the middle is a green island surrounded by a small canal, bordered by 2 rings of statues of famous personalities, a place where people study, chat and chill out. In Padua's historic centre, you will find anything your heart desires. Striking old buildings with impressive features, narrow, cosy streets, and again, statues. Right behind it, the famous Basilica of St. Anthony of Padua. St. Anthony is omnipresent because he is venerated as the city's patron. It is, quote, the most famous and artistically important architectural complex in the city of Padua. Off we go to the Palazzo della Ragione. Such picturesque daily markets and squares characterize this amazing city. And here it is, the famous Palazzo del Bo. Bo means ox. So, the Ox Palace has been the historic seat of the University of Padua since 1539. The Bible shows the way to go to heaven, not the way the heavens go. Once commented the famous and contentious Galileo Galilei, an outstanding lecturer. Most people today can only dream of his wealth of knowledge. This guy was an astronomer, physicist, mathematician, philosopher, and an engineer. In this aula magna, he taught mathematics. And now, please meet Dr. Elena Piscopia, the first woman in the world to earn a PhD. She lived and studied in the 17th century and became a respected philosopher and theologist. The University of Padua was founded way before this, in 1222. It is, therefore, one of the world's oldest universities. Today, it is a school of excellence, and the University of Padua has even brought forth several Nobel Prize winners.
Okay, so that's just yet another brief introduction about Padua. So we can go very long about Padua, and being as I mentioned, one of the alumni and lived there in that particular city for a number of years, I am always warm-hearted about that city. So, the conference would most likely be hosted at the Department of Biomedical Sciences, which is having a walking distance from the Padua city center, and many nearby hotels. Okay, and excellent value hotels, which is usually 25% cheaper than that of Milan on average, and 45% cheaper than Venice. So, if you would like to, let's say, spend few more days in Venice, Padua would probably be a better place to live in, because you'd save a lot of money for the accommodation. Okay, and in terms of the accommodation, as you can see that here, this being the Department of Biomedical Sciences, and there are plenty of hotels within, let's say, 500 meters of walking distance. So there are plenty of options that you can explore, many different hotels to spend your time, and all of these places are within the walking distance also to the train station, which would then directly take you to Venice if you want to go. Okay. So with this, well, in terms of the organization of the next BI22, so there are a number of organizers who will be joining in. So also from University of Padua and myself from UK and around the globe. And with this, I would just like to bring to your attention some important dates. So 31st December 2021 would be the deadline for special sessions, workshop and tutorials. 1st of February 2022 would be the paper submission deadline. 1st of April 2022 would be the paper acceptance notification due. 15th of April 2022 will be the final paper submission and early registration deadline. And as we mentioned, 15th to 17th July will be the BI 2022 in Padua, Italy. So at the end, I would invite you to experience the magic of Italy next year, hopefully. Okay, thank you very much for your attention. And well, if you have any question regarding any of the organizational stuff, if you would like to get involved, if you would like to contribute to the successful organization of this conference, please feel free and drop an email to any of these chairs that I have just shown. Also, particularly if you drop an email to myself and Dr. Yang present here, would be very happy to get back to you and bring you on board into the conference. Thank you again. Over to you, Professor Zong.
Thank you very much, Mufti, Jing, and Stefano. I really feel exciting. Really, hopefully you can join the next year. It's wonderful. Yeah, this conference. Yes, I mean both Italy, Padova, Italy, and also Queensland University. Australia. I mean, there is also another innovation after the pandemic. Yeah, of course, there's also hybrid conference. Yeah, also twin city, twin university. Yeah, also, I think every now the Brisbane, Australia is in July is winter. Yeah, winter, but it's not cold. I've been to many times in the July, but of course also the Padova is summer. I think it's really the trend also not only enjoying this high quality or technical academic program, but also enjoying the nature and the culture. Yeah. Finally, I would like to invite Shem to give the closing remarks. Shem, please.
Hello. Am I audible, Professor?
Yeah.
Yes.
Good morning, good afternoon, good evening. Brain Informatics Conference Series has created a premium research forum on brain informatics, which is a multidisciplinary research field where researchers from neuroscience, cognitive science, medicine, life science, data science, artificial intelligence and machine learning, neuroimaging, ICT, etc. field are collaborated together. In this meeting, we have seen researchers of all this domain have submitted their contribution, and we have also seen eight distinguished, keynote speakers shared their researches, and there were 11 workshop sessions as well as 3 special sessions. All these things enlightened us, and I have also seen many researchers join this session through Zoom and other social media, other platform, and they have actually made this session interactive through their question answer. And I would like to thank all of these keynote speakers, the session chair, for their help. And unfortunately, we missed, the on-site conference, networking session, coffee break due to this pandemic. Hope COVID-19 will say bye-bye and allow us to meet physically in BI 2022 in Padova. I would like to thank Professor Ning Zong, Professor Yang, Professor Michael, Professor Michaela, Professor Marco Giorgi, Professor Thao, Professor Francesco Moravito, Professor Gopi Krishna, and Professor Mufti Mahmood, and many other people who actually work hard to make this BI 2021 a successful. We have also seen our students, our volunteer, share the news related to this conference through their social media platform. I would like to thank them from my bottom of my heart. And we have also seen our session chairs manage the session. I mean, they have actually managed our time and everything. So also, I would like to thank them. And we believe we will meet physically in BI 2022, and you all submit your best work in this conference. And definitely, the upcoming conference will be one of the best events, because we have already heard that, the Padova University is going to celebrate its 800 years. So we would like to be a part of this event. So please join us and submit your best work. And thank you very much. Thank you for your help and cooperation. Thank you. Up to Ning Zong.
Okay, thanks, James. Okay, I really hope working with all of you together. Also, hope you can continually to contribute next year of Brain Informatics Conference for another big success. Also, see you next year at our Brain Informatics 2022 conference. Thank you very much. So this year's conference is closed. Thank you.
Thank you very much to everybody, and see you.
Thanks all for your contribution. Yeah, and an unparalleled success.
Thank you. Thanks, Mufti. This new video is really very good. We should use this new one also put on our next year conference website. Yes, thank you very much.
Yes. Yeah. We'll use this. Yeah.
Okay. See you. Bye bye.
See you. Bye bye.
Hope to see you.